Golgi apparatus, Golgi bodies, Golgi complex, structure of Golgi apparatus, Golgi bodies. These are membrane-bound cell organelles found in the cytoplasm of a eukaryotic cell, next to the endoplasmic reticulum and near the nucleus. Golgi bodies are supported together by cytoplasmic microtubules and held by a protein matrix. It is made up of flattened stacked pouches known as cisternae. These cisternae may be 4 to 10 in number for animal cell Golgi bodies, though some organisms like single-celled organisms have about 60 cisternae. They have 3 primary compartments known as cisternae nearest the endoplasmic reticulum, medial, central layers of cisternae, and the cisternae farthest from the endoplasmic reticulum. Animal cells have very few, 1 to 2, Golgi bodies, while plants have a few hundred. Functions of Golgi apparatus. Golgi bodies, their primary function is to transport, modify and pack proteins and lipids into the Golgi vesicles to deliver them to their target sites. Animal cells contain 1 or more Golgi bodies while plants have a few hundred. Cis and trans Golgi network make up the outer layer of cisternae at the cis and trans face, and they are responsible for sorting proteins and lipids received at the cis face and released by the trans face, by the Golgi bodies. The cis face collects the proteins and lipids of fused vesicles in clusters. The fused vesicles move along the microtubules through a specialized compartment known as the vesicular tubular cluster. This compartment is found between the endoplasmic reticulum and the Golgi apparatus. The vesicle clusters fuse with the cis-Golgi network, delivering the proteins and lipids into the cis face cisternae, and as they move from the cis face to the trans face, they get modified to functional units. These functional units get delivered to intracellular and extracellular components of the cell. Modification mechanisms include cleaving of oligosaccharides chains, attachment of sugar moieties of different side chains, adding fatty acids and or phosphate groups by phosphorylation, and or removing monosaccharides, e.g. the removal of the mannose moieties takes place in the cis and the medial cisternae, while adding of galactose takes place in the trans cisternae. Sorting of the modified proteins and lipids occurs in the trans Golgi network and packed into the trans vesicles, which then delivers them to the lysosomes or sometimes to the cell membrane for exocytosis. Assisted by ligands bound to receptors triggering fusion and protein secretion.